My name is Brad Lusk. I am one of the co-chairs for the American Association for the Advancement of Sciences, Science and Technology Policy Fellows STEAM Team. The STEAM team is really interested in engaging with the community. So three primary pillars of engagement are STEM and diversity, equity, and inclusion, outreach, science communication, so really making sure that we're able to communicate well, and also integrating the arts with the sciences. And really a major tenet of the STEAM team is to ensure that this communication that we're having is multi-directional. So we don't want it to be about the fellows conveying information out to the public. We want it to be as much about that as it is about the public conveying information to us and us being responsive to that and really kind of collaborating with our community in inventive and imaginative ways. So with that, I'll introduce Anna Dimitrio. So she is an award-winning, internationally renowned British artist who works with BioArt, sculpture, installation, and digital media to explore our relationship to infectious diseases, synthetic biology, and robotics. She has an extensive international exhibition profile. There are a number of museums that she has had her art placed at. I'll name a few: Z or ZKM, Ars Electronica, Beaux-Arts, the Picasso Museum, Philadelphia Science Center. The list goes on and on. She also has an exhibit coming up at MIT sometime early next year. I'm sure she'll be more than happy to tell us about that. She's also presented at TED. She did a TEDx talk that I was just watching right before this, actually, about how she loves bacteria. And that's kind of how I came across Anna. I actually met her at an art hub in Israel, where she was also talking about how she loves bacteria. And I remember getting a little piece of metal tin and putting an electroactive microorganism on it. And that stirred up a really good conversation. So anyway, with that, Anna, I'll turn it over to you. Take it away.
So yeah, I was going to basically introduce myself. So these are some of my roles. I have lots of different roles, some in science settings, some in art settings. Things like I'm artist in residence with something called the Modernising Medical Microbiology Project at the University of Oxford, and something called the National Collection of Type Cultures at Public Health England, which is the oldest collection, the longest running collection of pathogenic bacteria in the world. I also have research fellowships in Brighton and Sussex Medical School in global health, and at the University of Hertfordshire in computer science. And I'm working on a number of other projects. And I'm currently artist in residence with the Helmholtz Institute of Epigenetics and Stem Cells in Munich as well. So I'll mention that at the end. So I thought I'd tell you a little bit of background how I got into all this. I trained in fine art. The way science was taught when I was at school wasn't very appealing. I went to a girls' school, and they seemed to be quite fixated on stopping us getting pregnant. So they would tell us stuff about the reproductive system most of the time, and I didn't really enjoy that. So I kind of shied away from doing science. I carried on with physics, but biology, which is completely bizarre because I am obsessed with biology, that I didn't pursue at school. And then I went and did a degree in fine art and became interested again in some of these narratives around science, things like life extension and other rhetorics that are kind of things that we, you know, that kind of pervade science throughout history, these different methods that we apply to these various things. And it was around the time, and I'm going to show my age here, it was around the time that the World Wide Web was becoming accessible to ordinary people, such as art students, and you could start to look up things. I won't say Google because it's years before Google, but you could start to search for things and find out stuff that you wouldn't come across of your own accord, because we couldn't look things up before. And there was a university library, a very good library, but it was an art library, so it didn't have things about science. And so I started to kind of research this, and I found out these stories about microbiology and bacteria, particularly E. coli bacteria, that were so different to what we were told. So what you'd read about or see on the news, you'd hear about, you know, E. coli bacteria is a pathogen and it will kill you, it will give you food poisoning or something like that. But actually, what they didn't mention was actually a lot of E. coli is living there in your gut, part of something which we now call the microbiome. In those days, it was called the normal flora of the human body. And I started to get kind of interested in the difference between what the lay people get told and what the scientists know. So I kind of started to go and work with scientists and get them to teach me all the techniques that they use, and I do all the work hands-on myself with them, and just learn like that as an artist, and then make artworks that communicate some of their stories. So this is a very early work, and it's called Chair Flora and Bed Flora, and the chair is needle pointed with an image of the bacteria that I found on the chair that I cultured in the lab and looked at under the microscope. And the bed is this quite large scale crochet, quite abstract crochet based on the forms of the bacteria that I found on my bed. And my first collaboration I managed to get through Brighton and Sussex Medical School, and I started working with one of the professors there who was an expert in allergy. But after a little bit, she knew I was really interested in bacteria, and after a little while she came to me and she said, "Anna, I found you a microbiologist to work with, but don't do that thing you do where you come out with all your crazy ideas because you'll frighten him off." But I went to meet him, and within a little while he was actually showing me his paintings, and it turned out that he had this enormous kind of depth of research around the history of microbiology and all this stuff, and we've been working together ever since. And so this was one of the very early works. In about 2010 he became a partner he was at that time regional microbiologist for Public Health England, which means he was in charge of microbiology services for the whole south of England. And he was made partner on this project called Modernising Medical Microbiology, which was to implement whole genome sequencing of bacteria in the UK Health Service as a diagnostic method. And I'm just going to have a little bit of water because I'm losing my voice. And so one of the big issues of that time was Brighton Hospital. I live in Brighton on the south coast of the UK. The hospital there had very high rates of MRSA, or MRSA as you call it. And I started to work with the scientists who were studying that. And one of them, called James Price, said to me, "And when we grow it on agar, we have this special agar, and it grows this beautiful shade of denim blue." So I became fascinated by that, and I thought, well, maybe I could pattern fabric with the MRSA bacteria and then create kind of images on it, patterns, if you like, with antibiotics, so I could kind of control the growth. So MRSA is methicillin-resistant Staph aureus, so it means it can't be treated with the methicillin group of antibiotics. And so I tried out this experiment, and then obviously to take things out of the lab, you need to sterilize them. You can't exhibit a category 2 pathogen in an art gallery. So we needed to be sure we could sterilize it and kill it. And we went through the whole process and we managed to sterilize it, and it kept its blue, which was amazing. So I made this quite large scale work called the Mrsa Quilt, which has been shown all over the world. It's been to the States a few times. And you can see the different effects of different treatments, different tools and techniques in the treatment and diagnosis of MRSA. So you've got a sort of almost white square in the middle, which is vancomycin, as if it was an intravenous drip. You've got these polka dots, which are created by vancomycin susceptibility discs, which is a way that they test on Petri dishes on agar plates if the bacteria will grow up to the little disc of paper or not. You can see it in the other slide there, the little disc of paper, and then you have this, what they call a zone of inhibition around it. From that, they were also researching tuberculosis, and I became very fascinated in the history of TB. And this is a pneumothorax machine, which was a treatment for tuberculosis in around the 1950s, where they used to pierce the chest cavity and insert air into the chest cavity in order to collapse the lung. And I've carved the pneumothorax device with the images of lung tissue as if it was being destroyed by TB, and the cylinder in it is engraved with this, what they call the characteristic scattered red ribbons. And this device is something that's been tested again more recently against multi-drug resistant tuberculosis. And we're sort of used to the numbers of deaths for COVID per year now. So I should tell you that tuberculosis kills year on year, pretty much every year, between 1.2 million and 1.5 million. And we expect it to be higher last year and probably this year, because the symptoms are so similar to COVID in the early stages, and there's been very little diagnosis or treatment of TB. So in due course, this might be a treatment that actually gets brought back again. They've done trials, and it had some limited benefits for drug-resistant tuberculosis. This issue of antibiotic resistance is really important. The previous chief medical officer in England, Dame Sally Davis, said it was as big an existential threat to humanity as climate change. So to put that in perspective, it's a big issue. And I was really fascinated in how I could communicate the tools that they were using. And this story of MRSA, because we think of it as a recent thing. We think drug-resistant bacteria is new. But MRSA actually came out in 1960, was the first time it was documented. So I made this dress from a 1960 dress design, and it's patched in the same way as the quilt, but with the Staph aureus from my body, the methicillin-resistant Staph aureus, and the vancomycin-resistant Staph aureus. So that's Staph aureus, it means golden bunches of grapes, if you're wondering what Staph aureus means. That's just what it looks like under the microscope. And this bacteria, which is part of our normal skin flora, it lives on the human body, but it can attack us and cause disease. And so, yeah, this vancomycin stuff's the last ditch antibiotic that you would use if you had that infection. And I learned from the whole way through, I learned the Oxford University pipeline for whole genome sequencing. So from culturing the bacteria right up to doing the bioinformatics myself and assembling the genome. And when I'd assembled the genome, this 2.8 million bases of the Staph aureus genome from my body is projected as a kind of halo behind this. And the dress is video mapped with the light output of an Illumina whole genome sequencer as it's sequencing the infection. So the whole thing, it kind of communicates that whole genome sequencing is this light output, based on light output, using this method, and kind of revealing how they do that. And just for me to be able to really communicate it, I kind of need to know and do everything hands-on. And as part of that, the National Collection of Type Cultures, as I said, the oldest collection of pathogenic bacteria in the world, partly based at Colindale in London and partly based at Porton Down in Wiltshire. I've added to the collection the Staph aureus from my body, which I did whole genome sequence, so they've got that as well. And it's the first organism in their collection, which is vast and dates back to 1915, where they've got the world's oldest living cholera. They freeze dry them. So I learned the whole process of how to put something in the collection, and added my Staph aureus to the collection. And it's the first one in the collection for its artistic relevance. So you can look it up, and bioartists of the future, maybe they'll want to work with it. And if they do, it's NCTC 14139 is the code number. I've worked a lot with gut microbiota, and this, I can't go into these things very deeply because I know Bradley wants time for a conversation as well, but this is made in a gut model where they're investigating how Clostridium difficile infections grow. And some of these kind of pendulums here, the piece is called spindle because Clostridium means spindle. And these pendulums are grown very hard and dry. It's all made out of linen because Sergei Vinogradsky, who first discovered how linen is retted or rotted by bacteria, how it's flax, I should say, is rotted by bacteria in order to produce linen. He discovered that was due to Clostridia bacteria, these spindle-shaped organisms. There's no documentation to say whether he came up with the term Clostridium and whether he was inspired by the rest of the linen process. We don't know. But these are grown with the biofilms of C. diff, which are one of the reasons it's so difficult to treat. It causes kind of ulcerative kind of diarrhea in the gut, and you can die of it. And the only really useful treatment is a human microbiome transplant from a fecal sample from another donor with a healthy gut. The only other option is to take more antibiotics for it, which obviously, as it's caused by taking antibiotics, which creates dysbiosis in the gut and allows space for these Clostridium difficile bacteria to take over like weeds, that's not a brilliant solution. So I'm working a lot with scientists who are working on things like human microbiome transplants, and I've got a piece in Eden Project in Cornwall about that. Excuse me, got my cold still. This piece is called, it's from a series called Teeth Marks, and it's a collaboration I did with Birmingham Dental Hospital, and it's called The Most Profound Mystery, and it talks about the history of dentistry. Birmingham Dental Hospital in the UK is one of the oldest dental hospitals, if not the oldest dental hospital in the world. Back in the original days of it, it was built on the edges of the jewellery quarter there, which was the largest jewellery quarter in Europe. And I was telling the story with this piece about how people would carry fans to cover up their horrible teeth and bad breath in that era. And they were made out of bone, as this fan is, and they were fixed and repaired with gold wire, as this fan is. And then the silk on it is dyed with two different gum disease causing bacteria. You've got Provotella intermedia and Porphyromonas gingivalis. And I have to say, these are some of the smelliest bacteria I've ever worked with, which is strange because I work a lot with gut bacteria. Much worse though. I had to spray some air freshener on this little insight into my creative process here before I could exhibit this, because it was a very interesting work, olfactory wise. I became fascinated by the idea, could I heal antibiotic resistance or could I repair antibiotic resistance? When I sequenced the Staph aureus from my body, I discovered it was penicillin resistant. This is not uncommon. About 80% of Staph aureus is penicillin resistant. And I became fascinated. Could I do something like what they used to call plasmid curing, or something like that to remove this antibiotic resistance gene, which was on the genome of this thing. And I investigated lots of different ways of doing it. And in the end, I working with Technion in Haifa, which is when I was over there and met Bradley. I was able to take an E. coli bacteria that had an ampicillin resistance gene, which is related to penicillin, as part of the same family, and use CRISPR DNA editing, CRISPR gene editing, to remove the antibiotic resistance gene, which was on the genome of the E. coli, and repair the bacterial genome with the phrase make, do, and mend, which relates to this sort of wartime obsession with patching and repairing that everyone was kind of asked to do back in the World War II. And this was linked to the fact that Florian Chain, the scientists who created penicillin as a drug and won the Nobel Prize alongside Fleming for it, were working in Oxford in 1941. And there was also something that came out at that time called the Controlled Commodity Mark, CC41, that was stamped on clothes and furniture and anything that was using commodities well. So I was really interested in the relationship of clothes and furniture being controlled commodities, but antibiotics being used very liberally in such a way that we now have this very difficult situation. And so you can see the original, one of these original pamphlets there. And on the side is a piece that I made as kind of preliminary work to this Make, Do, and Mend series. And these silk squares here are grown on a chromogenic agar, which is color changing agar like I used before. But this one's called a UTI agar for urinary tract infections, but it's good for gut bacteria. And E. coli grow pink on it and Enterococcus grow blue on it. And what you kind of want for a nice, healthy, diverse gut microbiome would be a mixture of large and small pink and blue colonies. But as you can see here, some of the samples that we worked with are all overgrown with Enterococcus or all overgrown with E. coli. And that means that these people have got very undiverse gut microbiomes. And this is patients in a study at Oxford University into drug resistance in gut microbiota. And to do this project, just to give you an idea of how in depth I do go into all this, I had to have a typhoid vaccination because the bacteria, well, the fecal samples we were working with were from unknown sources. And so this is what I did in the lab. This is the first instantiation of this E. coli when we knew we'd finally got it there and we were happy we could call it this new strain, Make, Do, and Mend E. coli strain. And I worked with the scientists in the AMIT lab for the decipherment of genetic codes, particularly Sarah Goldberg and Rowie Amit there to make this work. But I did all the work hands-on myself with them supporting me. And the interesting thing is my experiment worked when quite a few of the PhD students in the lab, their experiments weren't working. The reason was I was on limited funding for my art. So instead of following the proper protocol for CRISPR, which was to pick 20 colonies and hope a colony had taken up the edit, I picked about 60 colonies, every single possible colony I could take, and then studied them, which obviously takes much longer. But one colony out of 60 had taken up the edit. And we know it's taken up the edit because we whole genome sequence, well, we sequenced it and sequenced it back out, and then we could see it was really there. And this is a suit that you can see, which was an actual CC41 controlled commodity 1941 suit. And I patched and repaired it with the silk that I'd grown with the CRISPR modified E. coli that I'd created. And there's different leaflets there about methods of patching and things like that, and the controlled commodity mark, and a tiny, tiny sewing machine, which was my mum's sewing machine that she used to play with during World War II, which is patching a tiny bit of cloth on the side there. And it made it onto the front cover of the CDC's Emerging Infectious Diseases journal. So that was nice. And it also made it to the UN General Assembly Global AMR Challenge event in New York in 2019 as well. And the Mrsa Quilt there. Not a brilliant photo, but I thought I'd show you for the fun of it. And I really go in depth into this, and I put myself in all sorts of strange and unusual settings. And this is me in a high security lab at Porton Down, working with Yersinia pestis, which is the bacterium that causes plague. So you can see some colonies of plague. These are these ones with these sort of pimples on the top. They're Yersinia pestis. And the other stuff is Staph aureus. And part of the test that they were teaching us was to tell the difference between Staph aureus and plague, which is a useful thing if you're a microbiologist. And this is in a category 3 lab in a hood under negative pressure, double gloved with gloves taped and everything. So it's really interesting. And I've written a lot about this notion of the bacterial sublime and kind of confronting these organisms that have literally changed the face of mankind. And I've been doing this for a long time. I think people now know that microorganisms can change the whole experience of humanity. The plague actually had a far higher death rate than the COVID-19 pandemic, so it caused incredible havoc. It killed half the population of China and a third of the population of Europe at one point. So it's been a very major thing. There's even still plague epidemics circulating now in places like Madagascar, and some of those strains have actually shown limited drug resistance as well, which is something to think about when we think about antibiotic resistance. And this is my plague dress, which is about this whole story, the story of plague coming from China through the Netherlands to the UK, the Great Plague of London. So this is a 1665 dress design, and it's stuffed full of all the herbs and spices and a lot of lavender that they used to use to ward off the plague, things like turmeric as well. And this embroidery on the front of the dress that you can see there is an actual period embroidery of the 1660s, and it's impregnated with the extracted DNA of plague that I extracted from killed plague in the lab with the scientists at Colindale in London, with the National Collection of Type Cultures. So it's perfectly safe killed plague DNA, but it's there on this dress, which is currently on show in the Rijksmuseum Boerhaave in Leiden, which used to be a pest house, a plague house for plague sufferers. So it's incredibly appropriate. And it also holds in its collection the most important microscopes of Van Leeuwenhoek, who was the first person to ever see bacteria, which he called animalcules, under the microscope. And I was very pleased that last summer, that the King of the Netherlands, who can be seen in this picture, went and opened the show and had a look at the plague dress. So that was fantastic. Works that we've been doing in the lockdown and in 2020 is this one. It's about groundbreaking research and modernising medical microbiology, where they've managed to do this massive data set of about 10,000 patients, and each patient in the study is represented by a little TB bacilli that you can see here in this environment, which is the alveoli and the bronchi and the lungs. And these 4 different colours here are 4 different antibiotics. And what they've done is they grew the bacteria from each patient in a 14-well plate in the presence of different antibiotics. And from that data analysis, they can now infer from the genome of a TB patient's infection which of the first 4 frontline antibiotics would treat it, which really reduces people being given the wrong antibiotics or antibiotics it might be resistant to or something like that. And it's a method that can be used for lots of different organisms or developed for lots of different organisms. So in the future, you can envisage this high level of targeting of antibiotics, but there's loads of work to be done. And so it's an installation, I should say, and you stand in the space and you can kind of guide the bacteria that cluster around you to the right antibiotic. So it's to tell that story. And these are some close-ups. And we also work with Gatwick Airport data, which is part of it as well, because we started the commission. I made the work with Alex May, as well as the Cryptic project at Oxford University. And we started the project before the pandemic started, and we were given access to Gatwick Airport's flight data, which we were correlating with the origins of the data that we were working with, and all the different organizations who were collaborating, because health is a global issue, which I've said for a very long time, and it's still just as true today. And we managed to have, by this unfortunate coincidence, all the data of the lockdown and coming out of the lockdowns as well, all from Gatwick Airport as well, which is layered into the piece. Also work with robotics, as Bradley mentioned, and this is one piece that's been shown quite widely around the world, which is called Archaea Bot, a post-singularity, post-climate change life form, and it's based on these ancient microorganisms called Archaea, particularly one from your neck of the woods, from Yellowstone Park. It's called Sophilobus acidocaldarius, and it lives in the hot springs at Yellowstone Park. But it's believed to be one of the oldest life forms on the planet, if not the oldest. And we were working with scientists on an EU project called the MARA project at Imperial College, who are studying the molecular motors that they use to drive these tails that they can use to move themselves around. And they were looking at them using electron cryo microscopy, and I went to work with the scientist who was doing that. And we got these kind of best models of the tails, of the molecular motors, and they went into this robot that we made, because the MARA project were talking about, can we make molecular robots using these motors? And their idea would be to take the cogwheel-like motors from the Archaea, sculpt them using a technique called DNA origami microscopically, and attach them to bacteria, replace the tails with drill bits, for example, and send these into the body to drill into bad cells that you didn't want anymore. So we're really fixated on this idea of robotics and these ancient life forms who love hot conditions, and they love acid. They love acidic conditions. They wouldn't be bothered by acid rain. They'd be very happy with this sort of situation. So we kind of hypothesized about this sort of fear of humanity, of climate change, and also the fear of AI. We made this in about 2017, and that was quite a hot topic. 2018 as well, quite a hot topic then, the fear of AI and the coming singularity. If it is indeed coming, that could be a topic to argue over. And so this has a neural net in it. It's sensing its environment and learning about its environment, and the tails are reaching around, and it's kind of a sort of dystopian solution for climate change, a kind of with a view that maybe you could upload your consciousness and live on in a kind of robotic Archaea form in the future. It's kind of a hypothetical work, but also communicating what Archaea are, what they're like, that some are extremophiles, and all these other stories around it. And we've just finished a work which has just gone on show in the Museo Bolivariano in Santa Marta, Colombia, which we launched a couple of days ago, which is a video version of it. And the Archaea are under the seas, carrying the last vestiges of human consciousness and kind of swimming through these ruins that are now covered up by the rising tides. There's a few images of it there. Just before the lockdown in 2020, we were in, with Alex May as well, in Switzerland working with Schindler, who you won't know, but they're nearly as big as Otis, who make elevators. They're like the European version of that. So they're like the second biggest elevator company in the world. And so we're working with this huge engineering company who are very future thinking and working at the very cutting edge of transportation stuff. So we made this robot called Cyberspecies Proximity Robot that was kind of playing with different ethical concerns around the future of robotics and delivery robots. And the tiny little robot figure, it uses all the same technology as a delivery robot, but it can react to your body language. So it kind of pulls at your heartstrings in a way. And it looks quite like a worker, delivery worker. So it's kind of alluding to those things. A piece I made during the lockdown was about the impact of COVID-19 lockdown on domestic violence. And the piece alludes to Virginia Woolf's famous text, A Room of One's Own, where she said that you need a safe space, a room with a lock on the door, and an income of, I think it was £5 a year in her case. But she lived very near to where I live. So she was very much in my mind, this idea that to be safe. But during the pandemic, people who were subject to domestic violence were actually locked at home with partners who were abusive to them. So they didn't have that safe space. And so the installation takes the form of a kind of temporary hospital, but with these handmade bedding and stuff on it. And the pillows are impregnated with the extracted RNA of SARS-CoV-2 from a plasmid construct. So it's a safe reagent, but it is the RNA that causes the disease, or based on that anyway. It's from a plasmid construct, so it's created in the lab version. And so this is completely safe, but it's on the installation, again, to give that kind of frisson of the real stuff being there. And it's quite the beds are exactly based on models from the hospitals in Wuhan, from the first emergency hospitals, and the beddings dyed with different natural dyes that are used to treat infectious diseases. And as you can see, it's quite a tiny installation, so that's me setting it up there. This is what Bradley was talking about, this piece, ex voto, and I collect people's stories about the impact of infectious diseases, mainly bacteria and antibiotics, on their lives, and create this installation with it. And all the ribbons also tell their own stories. They're either dyed with antibiotics or they're dyed with different things. And that was it in the what's it called? The Valkhof Museum in the Netherlands. Most recently, I've been in the lab in Vienna, in Boku, working with the scientists there, and we've created this yeast, working again with Alex May, and the yeast can capture carbon and output lactic acid, which can be polymerized to make PLA. So it's like it solves the carbon problem, and we can have as much plastic as we want. So that's the sort of thing artists come up with. Lactic acid is very toxic to the yeast, so it needs to be evolved in the presence of lactic acid many, many, many generations before it's going to be a useful solution, but it does work. And this is another work we made called Culture, that's about the co-evolution of yeast and human culture, and about the beauty of yeast colonies and how they're considered kind of damaging to statues and antiquities like these things. And this is a collaboration with the University of Western Attica, where I've been basically investigating the nature of conservation and the conservation of bioart, and also using blockchain technology, which is why they've got these RFID tags embedded in them. And then most recently, this piece is at the Kunsthal Charlottenburg in Copenhagen, and it's about future medicine. It's a very complicated artwork, so I would direct you to my website for the key to what's in all the boxes. Suffice to say, I've got things like carbon capturing yeasts, transposable elements, jumping genes, all sorts of things. Oh, and most importantly, a psychobiotic, a plasmid that can enhance serotonin production in the gut. And my latest project is called Collateral Effects, and I'm working with loads of scientists from like the Wellcome Sanger, Public Health England, Oxford University, Brighton Sussex Medical School, Birmingham Dental Hospital, and Future Everything, to explore the collateral effects of the pandemic, specifically on antibiotic resistance, but also on the nature of scientific research. So that's like literally a whistle-stop tour. The latest thing I've been doing is staining cells at the Helmholtz Institute, in the Institute for Epigenetics and Stem Cells. So I'm on a steep learning curve at the moment, dyeing cells and looking at them under fluorescence microscopes and exploring things like heterochromatin and euchromatin and cell memory and cell fates. But that's coming next. That's only in the early stages. And that's all my supporters. And that's how to keep in touch with me. So I think I've given you a real whistle-stop tour, and I'm happy to take questions and things like that now.
So if anyone wants to, raise their hand or what have you, I can call on you and you can ask a question. I'll go ahead and get us started with a question that struck me while I was watching, and that is, you got MRSA and plague and C. diff and a lot of these bacteria that are really associated with kind of disease and infection, and no doubt have kind of altered human history. Mm-hmm. I'm curious, why have you chosen kind of those maybe more infectious agents as opposed to some other ones? Although I did appreciate your Archaea at the end, and as someone who studies thermophiles, I very much appreciate that. But yeah, just maybe a little bit more about why you chose some of those more nasty bugs.
Well, I mean, they're the most important ones to us. They're, they have literally changed the face of human history, and they affect people on a daily basis. And the amount of people who come up and want to know more because either they're affected or somebody else that they know is affected by those things, it's really important to work on that, and not many people do. And I think that's because it's quite a big undertaking. You have to be quite into it, I think, to really learn all that stuff and do it, to make the work that that focused on it. Otherwise, you just tell a superficial story, which is not something I want to do.
Okay. And we have a question here from Elizabeth.
How do I sterilize them? Well, it depends. So DNA is sterile, so it doesn't have the infectious agent in it once you make sure that it is the DNA only that you're using. So there's ways you can do that. There are kill protocols that you can go through where you try and repeatedly grow bacteria from it to ensure your protocol's working perfectly. So this was the one that was approved by Oxford University that we used. In fact, my collaborators developed it. And for the tuberculosis, it's all different for each bacteria. But so Clostridium difficile biofilms, you can autoclave, but at a very high temperature on a kill cycle that you're never gonna get it back usually. So people don't usually take it back at that point. But it's the way that if you were going to send it to like landfill, if it was going to go to the rubbish dump, the garbage tip, as you might call it in America, I guess. Yeah, that's how you would do that. Then for the things like the MRSA, that's autoclaved again. So you just put it in a big machine and sterilize it under high steam pressure, and it's dead. So it's the same way that basically they sterilize things in hospitals. I usually use hospital autoclaves.
I appreciate it. Thank you. And thank you for presenting to us today.
Thank you.
I'm kind of curious too, as most of us are scientists in various fields, and a big part of what the STEAM team does is figure out ways that we as scientists can communicate messages about things like what you're communicating, infectious diseases, how people should act or not act during the COVID-19 pandemic, things like that. Also ideas of there's different sources of information out there, and how do we convey that these sources are reliable and these ones aren't. And I'm just curious if you, as an artist with this kind of really innovative and kind of cool way of communicating about science to members of the community, kind of what what advice or like what best practices do you use when it comes to like conveying this, or is there any like cool, insightful things that you've come across while having discussions about science in this format?
Well, I mean, I think most people can understand stuff if it's explained in a way that sort of promotes their understanding. And that doesn't mean dumbing it down. That means like directly engaging. So one thing that we do a lot is we do kind of participatory stuff. So with the Mrsa Quilt, we actually did an Mrsa quilt making workshop at the Victoria and Albert Museum in London. So we had our Petri dishes of that special chromogenic agar, and people had a selection of different antimicrobial and antibiotic things, and they could make their own quilt squares and then lay them into the Petri dish. Whilst they were doing that, they could have a conversation with the scientists who were also sitting there stitching and making these things. So there was no hierarchy. I mean, they didn't know they were talking to the guy who was in charge of microbiology for the whole region. You know, he was by that time London and the southeast. He was in charge of. So like they didn't know, so they could just have like little conversations and just chat. And you know, and he's really nice, so like he wouldn't be, you know, any kind of pompous or anything like that. So it was like really like sweet, and people would have like really good conversations. And you can ask a question in a little chatty making session much more easily you can than a big sort of, you know, if you go to a science festival and you want to be the person that asks the question at the end, you've got to put your hand up and you're like, excuse me, can I ask a stupid question? No, you're not going to ask a stupid question. You're only ever going to ask a question that makes you sound clever at one of those things. So people don't get to ask stuff. So it's just like opening those things. And then when I show the works, I usually show them with a little paragraph of text. And if it's a particularly complicated work, then maybe another paragraph and another paragraph. But you could get away with just reading the top paragraph if you didn't want to do a load of reading, you know. So, and my friend Nicola is one of the collaborators on the work. She said when I had the show at Oxford Museum of History of Science, because I had a solo show there, and she said she would love following people around the show. And they'd sort of do this, they'd sort of peer at the artwork and go, and then they'd look at the sign, and then they go, oh, and then they'd peer back at the art, and then look at the sign again, and things like that. And she said she loved it, you know, like watching that realization of people. And then they wanted more information, and then they would come to the talks that we provided, or they could ask the gallery invigilators who were trained on the work and stuff as well. So I mean, that was quite fun because they put a great big sign outside the museum, which is in Broad Street. If anybody knows Oxford, you know it's the dead centre of town next to the Sheldonian Theatre. And they put a great big sign on the banners, and it said BioArt and Bacteria, the art of Anna Dimitrio or something. And like, apparently loads of people just came in because they didn't know what BioArt was, and they were fascinated. And it had huge visitor numbers, so it was fantastic, you know, just that. So people are really interested in it if they get a chance to see it and experience it. And then we'd have like kids coming to the workshops, and they would just come back, and they loved it. And there was this one little girl, she was about 8, and her mum was like, what, or one of the museum staff I think said, what do you want to be when you grow up? And she said, a synthetic biologist. So that's good, isn't it? Because she'd been coming to this, you know, my workshops and things. So, you know, all that stuff's really nice. I think it's just like really breaking down those barriers and chatting with people and just making the work. It's complicated work, but try and make it as accessible as possible.
Yeah, that's all great to hear. It sounds like kind of using art and inviting people to the table is almost a way of democratizing the conversation and enabling people to be curious and to just wonder and to maybe be more open to asking questions that otherwise they might be intimidated to ask.
My question is whether when you approach how to present a bacteria, whether there's a difference between if it's a bacteria that's always with us, like C. diff is always there, and it's when it runs rampant that it's a problem, versus something like plague where it's a problem once it's introduced.
I mean, I go into the story of each of the bacteria. So I mean, I don't know that it's strictly true that it's always with us all the time, C. diff, because there's a lot of people that don't carry it at all. It's very common in babies, but also we don't know if that's got the toxin, if that's toxin producing, if that's actually causing illness in the babies. And actually, my collaborator's studying this, so to push that. But in older people, it's not all the time in people. It's just that if they are carrying it and then they take antibiotics, then you get this issue of this dysbiosis where it takes over. So, I mean, I just try and make works that tell the story of the organisms and the researchers that I'm kind of working with. So, I mean, in the case of the plague stuff, it was about the DNA, really, because this was part of a big project called the NCTC 3000 project, where they used biosequencing to sequence 3,000 of their most famous organisms at the Wellcome Sanger Institute. So so it was part of that kind of story there, the sort of cutting edge. And then it was originally launched in the Guangzhou Triennial in Guangzhou, which used to be called Canton and was the largest maritime port on the Silk Road, which is the sort of the route that the plague took. So it's sort of telling these stories as well. So with each of the things, I don't know if there's a difference in how I feel about them. I mean, they're all different, and I try and tell their stories.
You talking about this and the Silk Road and this kind of tie between maybe globalization and commerce, and like the spread of these infectious diseases that comes along with that, and kind of just weighing like the good and the bad in terms of like, yeah, we have access to economy, but we also have these other things that might come with them. It's just an idea that just came to my head now as you were talking.
I mean, the piece is alluding to that story of like the journey of the disease, but there's nothing in my work that says that we shouldn't trade and spread or whatever and trade like that, because I'm not a great believer in that. I mean, disease has been going on since man, you know, first kind of moved from, you know, living in the trees to living on the ground. And then when we started to form, rather than hunting and gathering, we started to kind of form into groups and things, you know, live in huts in close proximity with animals. You know, it's this stuff, the ships sailed on all that. So I think, I mean, now we've got amazing technologies that you can come up with a vaccine within, you know, months of a pandemic starting. They were literally in Oxford, they were literally in production mode by about April, you know, because they had the whole thing kind of set up, ready to plug in a disease to it, to their method. So, you know, to go from that to that, it's, you know, it's incredible, really. So yeah, I'm not saying that we shouldn't travel or be global, but we've got to find good ways to do it, like more, you know, carbon neutral travel and things like that, better aeroplanes. But I don't think it's very helpful, like, just to say none of us should travel anymore, because traveling is what really opens up your mind, isn't it? And you know, meeting different cultures and stuff, it stops that kind of idea of, you know, them and us kind of thing. So I think everyone's should travel. It's really important.
Yeah, I agree. I agree. It is just an interesting double-edged sword to think about. All right, so with that, if there are no other questions, I'll thank you, Anna. I appreciate you showing up, having this conversation with us.